I welcome everyone on hands-on workshop on advanced Drosophila genetics. The fruit fly Drosophila is conventionally associated with genetics because of the dominance of post-Mendelian genetics since 1910. By studies that is used, it is famously considered as Cinderella of genetics. During the past century, the subject of genetics has evolved and has become central to all areas in biology. It is significant that during this period, Drosophila has also become a favorite model for studies in almost all aspects of biology, including human diseases and personalized medicines. On this note, I request our beloved chairperson to welcome this auspicious gathering. Now I request Ms. Manonmai for the invocation.
हम सवाहिनी ज्ञानदायिनी हम सवाहिनी ज्ञानदायिनी ज्ञान के भंडार दे ज्ञान के भंडार दे शारदे वरदान दे शारदे वरदान दे हम सवाहिनी ज्ञानदायिनी जो विधात्री यो विधात्री जो विधात्री यो विधात्री ज्योति का संतान दे ज्योति का संतान दे शारदे वरदान दे शारदे वरदान दे हम सवाहिनी ज्ञानदायिनी विश्व के अंधेर घट में गिर ना जाए इस सफर में दीपों का हार दे दीपों का हार दे शारदे वरदान दे शारदे वरदान दे हम सवाहिनी ज्ञानदायिनी ज्ञानदायिनी.
Now I request our chairperson, Professor S.S. Malini, for the welcome speech.
A very good morning to all of you. A warm welcome to all the dignitaries, participants, students, staff, everyone. I take this opportunity to brief about the scope of this workshop, as well as I welcome all of you. The fruit fly, Drosophila melanogaster, is a versatile model organism that has been used in biomedical research for over a century to study a broad range of phenomena. There are many technical advantages of using Drosophila over other vertebrate models, and it is easy to handle, inexpensive to culture in the laboratory condition, and have much shorter life cycle, and they produce large number of progeny, that's externally laid embryos, and they can be genetically modified in numerous ways. Research using Drosophila has made a key advances in our understanding of regenerative biomedicine or the biology, and been successfully contributed in the field of regenerative medicine in many different ways. The relationship between fruit fly Drosophila and human genes is very close, that often the sequence of newly discovered human genes, including disease genes, can be matched with, it is equivalent to genes in fly. 75% of the genes that causes diseases in human are also found in fruit fly. So experimental studies are very essential learning tools for the students of biology, beside providing better understanding and appreciation of what they learned, that is a theoretical part, through reading and through lectures. Experimental studies provide hands-on experiences and expertise also, reinforcing the learning process and arouse curiosity among students. With the astonishing progress in biological sciences during the past several decades, the domain of experimental work has also substantially expanded. Drosophila research in the portal of University of Mysore commenced in the Department of Zoology under the stewardship of Professor M.R. Rajshekhar Shetty. After returning from Columbia University, and it's my pleasure to inform you that he's a student of Dobzhansky. And later on, many others, eminent scholars who have trained under famous genetics group, Muller and Painter, joined the department, and they have started the Drosophila research, and it went on uninterruptedly. And among the progenitors of researchers, some of them, Professor Sri Ram Reddy, Professor Siddha Viragowda, Professor H. A. Ranganath, Professor S. R. Ramesh, Professor V. Vasudev, Professor V. A. Vijayan, Professor N. B. Ramachandra, Professor Shamala, Dr. Shakuntala and Krishna. Among them, Professor H. A. Ranganath pioneered in commencing the renowned Drosophila Stock Center in University of Mysore along with his colleagues. And he was instrumental to bring two national research facilities that at the University of Mysore. One is Drosophila Stock Center and another is Unit on Evolution and Genetics, funded by DBT and DST respectively. And he was a former NAC director, former VC of Bangalore University. Professor N. B. Ramachandra and his students, he's a first student of H.R. Ranganath sir, and his group recently worked on whole genome sequencing and gonadal transcriptome of few cyteraces and was submitted to NCBI, and they have 26 submitted to NCBI. And it is a great pleasure to inform you that today's keynote speaker, Professor Upendra, he is related to this lineage, who is the student of Professor N. B. Ramachandra, first student of N. B. Ramachandra sir. And at present, he is working as a professor in IIC Bangalore. And his specialization is once again genetics of Drosophila. So today, to grace this occasion, we have eminent personalities with us, and a man of high vision, great scholar, educationist is none other than our Vice Chancellor, sir, Professor G. Hemant Kumar, sir. Sir is alumni of our university, and he has occupied many responsibilities at different capacities in the Department of Computer Science as well as in the university. And he is instrumental in bringing Career Hub in university and also Engineering College in Mysore University. He has mobilized more than 10 crores grants from China with the MOU with Mysore University. And more than that, he's a great teacher and a great human. So on behalf of Department of Genetics and Genomics, and from all of us, a warm welcome you, sir, to this great occasion. And I take this opportunity to welcome Professor Upendra from IIC, and he's alumni of our Department of Zoology and first student of Professor N. V. Ramachandra. And he did his BSc and MSc from Mysore University. And 95 to 99, he pursued his PhD in zoology department under the guideship of Professor N. V. Ramachandra sir. And he worked on genetic analysis of indirect flight muscle development in Drosophila melanogaster. In 98 to 2004, he worked as a postdoc fellow in University of York. And in 2004, he came back to India and he joined IIC, Department of MRDG in IIC, Bangalore. And he continued his research. And in 2014, he promoted as associate professor. And in 2020, at present, he is working as a professor in the Department of MRDG, IISc Bangalore. And he is also adjunct professor in the University of Transdisciplinary Health Sciences and Technology, Bangalore. And to his credential, he has many affiliation, member of British Development Biological Society, life member of Society of Biological Chemists, life member of Indian Society of Cell Biology, and many more. And he has published many articles in highly reputed journals, more than 60 articles, and wrote a book chapter, one book chapter, then review articles, conference proceedings, and has got the highest citation, 784 is the highest citation, and 18 H index he has. And he has guided 12 students for his PhD. And on behalf of everyone, I welcome you to your alma mater again, home town. On behalf of everyone, a warm welcome to Professor Upendra. Now I welcome Professor N.B. Ramachandra sir, founder chairperson of the Department of Genetics and Genomics. Yeah, I welcome Professor G. Subramania sir. And I welcome Dr. Amritavalli madam. They are my strength in the department, and they are guiding me in all my activities. And it is my duty to welcome all young mind, young faculties in our department. Dr. Tejas, Dr. Pratibha, Dr. Chaitra, Dr. Somana, Dr. Kaushik, Manjunath, and they are instrumental to this program. Without their effort, we would have not organized this program. I welcome all of them. And I welcome a warm welcome to all the participants from different parts of the country. And students are from Gujarat, Tamil Nadu, Karnataka, Andhra, and we have limited the number. So even we were getting a lot of queries again, but we have restricted the number to 24 because it's a hands-on training. Otherwise, we'll fail in our duties if we are not giving proper training. So we have restricted, and they are all the way to Mysore once again. So warm welcome to all of you. I welcome all the students, research scholars, and non-teaching staff, and faculty, people from personals from ICD and IOE and all. So once again, a warm welcome to all of you. Thank you.
On behalf of everyone, I welcome our chairperson, Professor S.S. Malini. I request our Honorable Vice Chancellor, Professor Hemant Kumar sir, for the inaugural speech. So in order to keeping the wave of knowledge constantly active through the medium of flame, I request all our dignitaries to light the lamp and inaugurate this function. Now I request VC for the inaugural speech.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. Dignitaries on the dais, Professor Upendra, MRDG, Indian Institute of Science, Professor S.S. Malini, Professor and Chairman of Department of Genetics and Genomics, Professor Ramchandra, Professor Subramaniam, Professor Amrutavalli, and all other teachers of this genetics department and other departments, the students of this department, research scholars of this department, and research scholars and students coming from different parts of the India for this workshop. I'm very happy to inaugurate this 4 days workshop on advanced Drosophila genetics, which has been organized by Department of Genetics and Genomics. I'm very happy to inaugurate this program. So there are different participants and there was demand for this workshop. So this workshop has been designed for Drosophila genetics. As we are all aware that Drosophila, which is a tiny fruitfully, which is very commonly seen in their house, fruits on fruits and vegetables. I know it is famous as Drosophila all over the world. The main source of attraction of this 4 days workshop is in this department. Main reason is this is because well-proved bisexual eukaryotic organism which simulates many aspects of the biology, namely developmental biology, physiology, molecular genetics, molecular biochemistry related to the higher mammals. It is surprising that very recently it has emerged as an important laboratory model organism in the COVID-19 research. Using the computation analysis and bioinformatic tools, it is found that 90% of this of the virus host interaction involves with the human protein and conserved in the Drosophila. All you are aware that the Department of Studies of Genetics and Genomics is blessed by the strong school of Drosophila genetics in our country. It is noteworthy to remember the great contributions made by the faculties of the parent department in the field of Drosophila, that is Department of Geology in the campus. It is not important that they are part of the curriculum in the Indian universities at graduate and postgraduate level to teach the basic genetics, studying what we call biology of Drosophila. But recent progress made in the fly research is commendable and enriching the knowledge of the mentors, teachers in the recent techniques and advancement in the field of fly genetics is of the immense importance to train the younger generation. The department has a credit in releasing the genomics of what we call genomics of cytorays in NBCI. I have gone through the workshop, the highlights and its highlights. It is very interesting for me, I'm also specialized in computer science, because of this computer science research and also a lot of software tools that has been designed, because computer science evolved, took the domain knowledge of various subjects and what we call evolved the software. That software is saving your time and also giving what we call the accurate results. So that is what today's workshop is making all the new researchers to work on these softwares. I feel well-trained faculty of this department are capable of training the participants from orthodox to contemporary genetics of Drosophila. So I heard from the chairman that there is a huge demand for this workshop. That's what she told in her what we call speech also. The various faculties of the various different universities from south to north. I am confident this department has conducted many workshops like this and led this workshop in the regular intervals for the teachers at PG and also UG level. So you can also coordinate with the academic staff college for a 1-week program through them, or by our RUSA funding is there for this HRDC. So through that also it is possible to do it. Use that because we have got some funds and grants in that. This type of workshop, we can make it for 1 week and hands-on experience and use that, and let this department collaborate with HRDC and RUSA funds and let it go what we call for the young researchers, where this type of what we call research will lead you the further what we call different solutions to the nature and also to the different solutions that problems that we have in our country. So because any research should always reach the society. So this type of research really touches the society. So for that, we need to train what we call our students and researchers in a proper direction. So you have got that potentiality, collaborate with HRDC and other what we call resources in our university, because now government is not further funding to any of the what we call department. What our education department says, that generate the money, run the show. So for that, we must be prepared. At least not now, at least for 5 years, we had to plan how to run our department, how to run our these things, how to run, because once upon a time, I had 5 crores only for seminars and what we call conducting the seminars and workshops. So I used to call the different departments, conduct this workshop, because I had to what we call give the accountability for that. Now it is not so. No further funding from the central government and also the state government. We have to develop, we have to sustain on our own with this this department, and our science departments are all, we should motivate our research, our young researchers to in a proper direction to create something for the next generation. Thank you very much.
Thank you so much, sir. Since Hemant sir has other commitments, so he'll be leaving us now. So I request our chairperson to present a token of gratitude. Also request our chairperson to present the token to the keynote speaker, Dr. Upendra sir. Thank you, ma'am. Thank you, sir. Now I request our keynote speaker, Dr. Upendra Nongtomba, to deliver his keynote address.
Yeah, that's what I mean. Carry on. Well, it's my privilege to propose a vote of thanks. So at the outset, I'd like to thank our Vice Chancellor, Professor Hemant Kumar, for gracing this occasion and inaugurating the Drosophila workshop. I also thank him for extending his support in conducting this workshop. A very big thank you to our keynote speaker, Professor Upendra, for taking the time to come all the way from Bangalore to participate in the program. We are all excited and looking forward to listening to his keynote lecture. I would like to thank the participants who have registered to participate in this workshop. They're the primary people that make this workshop happen. I would like to thank members of the genetics department who worked hard and put together this workshop, which will undoubtedly benefit all our participants. Thanks to our chairman, Professor S.S. Malini, faculty, staff, research scholars, and students of the department who have worked toward making this workshop possible. A special thanks to the Drosophila Stock Center staff from the Department of Zoology for providing Drosophila strains. Thank you to Vignan Bhavan for making it convenient for us to host the inaugural function in their auditorium. Thank you, one and all. Thank you.
Yeah, now I feel comfortable, right? Yeah, so that I'm not bound by that there is another program after me, right? So I can take as much as I want, right? Okay, yeah. Sorry. So idea is you can interact with me, okay? So it's not be like one way bombarding information, right? So, and I'm not going to give you how Drosophila has been a wonderful model organism and how much we know from the Drosophila. The idea for me is to give you few concepts, the paradigm shift concept, which Drosophila has contributed. Then if you get convinced, you talk to me so that I can explain you more, right? That's the idea. So first of all, thanks to Malini and all the organizers for this workshop, for giving me this opportunity to interact with all of you. And I hope that this workshop is a successful one. And all the participants, you learn something from next 2-3 days, right? And so that when you go back, you can show not only the certificate of your participation, but also you can say that, you know, you learned something, yeah, for your lifetime, not for few days, but for your lifetime. So okay, this work, right? So my title will be nothing more, Cinderella of genetics, a versatile model to dissect basic concepts to precision medicine. So you know it, that how I'm going to convince you that how Drosophila was useful in making sense of everything. That is how we are here today, right? You all know that we all wouldn't be here if we didn't know how we developed, right? So that is the fundamental questions, right? So if you look at, if we were just a single cell, right? Like say amoeba, right? So if you look at the amoeba single cell, if there was no evolution, we would have been an adult human has close to say 120 to anything about 120 to 170 trillion cells, right? So we would have been amoeba with the 170 trillion cells, right? Mass of cells, right? So we are not mass of cells. We are different. We have head here. We have leg down there. If somebody has got the leg up, then I don't know, right? Yeah, but it happens. It happens in Drosophila, right? There is a mutation called antennapedia, where in the head region, you have a leg. But by knowing such a simple thing that the leg can be developed at the antenna regions, then the people ask that how it is possible, right? Mechanisms that, you know, how antennapedia is regulated, how it is down regulated, or they say simple things I don't want to make it too scientific talk, okay? So what I'm trying to convince you that you have 2 eyes, 2 ears, 1 nose. If somebody has got 2 nose, then let me know, right? Yeah, it happens, right? You can have 3 eyes, right? Like the Shivji, right? Bhagwan Shivji. You can have 3 eyes whenever you want it. Drosophila made more than Shivji. Drosophila people activated a gene called as the eyeless, and they could make eye all over body. It means that every cell has got the ability, because all the cell in your body is made from the same kind of genome. So they have the ability to make eyes or ears or whatever you want it in the every part of your body. But the only thing is that you have only 2 eyes where they are supposed to be. We all have 2 eyes, right? Because in the other part of the body, your eye making genes are repressed. Now the question comes is that how the eyeless gene or PEC6 in the human, right, the autolog of eyeless, the master regulator we call it, right, something which express it, activate it, if they can form the whole organs or the tissue, such a gene cascade or such a gene is called as a master regulator. Okay, so eyeless is a master regulator. Now, you want to know that why these things are happening, right? And I will come back to you. This basic thing you need to know, and the basic thing we need to know for the every gene which are there in our body or even in the Drosophila, right? If you want to understand why we develop diseases and how we can cure it, right? Okay, so that's it. So don't get bored, right? Yeah. It's the same thing as the plants. You don't fit the flower in the down, right? Yeah. So there are, you know, the gene name, plant name, or something is different, but ultimately what in the development, the context is that how development take place. Most importantly, say from the single cell to individual, like this guy who is enjoying the nature, right? Okay, because if I was just like this, then I will not be crawling around here in the water, right? It would be something other different organism who is used to the crawling in the water, right? A fish or say amoeba or something, not this organism, right? So how these multicellular organisms with the right proportions, right proportion is also important, because you cannot have very long hands, limitations are there. You can't have a very long leg which is not proportional to your body. So what dictates all these things, right? Okay, that basic question started with Drosophila, right? All these questions in the developmental biology, if you look at, if you really want to understand, you are byproduct of all these things happening in the body. Of course, I haven't added one here, the presenting what we call as the biomechanics. Okay, I haven't added it now. The biomechanics is very important in development today. Okay, all right. So I'll give you a few examples, maybe if time permits. So you need to choose if you really want to know, you know, what you want to do with the development. If the whole organism development, or the say eye development, or the say ear development, whatever you want to choose, it is a complex, remember, all right? Okay, so it is the same thing actually. You can take any organs. What you are doing is that if you work with the eye development, eye is part of the body. Okay, you will have a signaling cascade which is making it eye, but ultimately it is same as like a human, I mean the whole body. Okay, because the signaling will be different for the different organs, that's all. Okay, different tissue. But the process will be like switch on, switch off, something happening with the interaction with the, say, neighboring cells or the environment. That is what you are going to look at, right? Okay, and that is what you need to look at. So depending on what is your question, you have to choose the model organism. The principal thing in knowing the model organism, choosing the model organism in a place like India is that, say if you want to look at how we call it as the human disease, let's say actinopathies, the actin aggregate or the protein aggregate, which is responsible which arise from the mutation in the actin, you don't have to choose any higher model organisms. You can actually choose single cell organism called Dictyostelium. Dictyostelium, if you take it, they have the same kind of actin. That is not much different. Actin is one of the most conserved cytoskeleton protein. Okay, between Drosophila and the human, there is only 6 residues which are different, right? So you can use Dictyostelium. You don't need anything to work with the Dictyostelium. You don't need even money. Okay, just like Vice Chancellor said now that there will not be any money. So no question of working with the mice or any other higher organism, even Drosophila. Sorry to say it, Drosophila also require money, okay, for making food. But if you work with the Dictyostelium, then you can just collect it from the soil everywhere and you can start working. But the problem with the Dictyostelium is that you will not be able to understand how eye develops, right? Yeah, how the ear develops, how other things which are not present, organ development, you will not be able to understand it. Okay, so you need to choose the right model organism which not be very expensive, which can be manipulated easily, right? And what you want to understand, it should be present in that organism. All right, that is why people started with Drosophila, and why Drosophila has been and would be still a very versatile model organism is because it is amenable to not only genetic manipulation, but in a place like India, Drosophila is not animal. You see, according to the Maneka Gandhi's Animal Ethics Committee, Drosophila is not animal. Okay, so you don't need Animal Ethics Committee clearance to work with Drosophila. All right. Okay, if you work with a higher organism like say mice or say any rodents or even fish, if you want to work with zebrafish, now you need to get the ethical clearance. But if you want to work with the Drosophila, thanks to Maneka Gandhi, because Maneka Gandhi never thought that fruit fly could be animal, right? Okay, so you don't need any ethical clearance. The best part is that because it is sequenced, unlike any other organism, all the mutations for the expected genes, right, are present, available from the stock center in all over. I mean, there are multiple stock centers, including Mysore. But if you want any stock center from any part of the world, you can get it. There you need money. For example, say if you want to get the say fly from Bloomington Stock Center, you have to pay $26. If you want to get the say RNAi sequence for any gene from the VDRC, we call it, where 44,000 RNAis are available for 1,400 and 14,600 genes, you have to pay $127. Okay, which is much cheaper compared to say getting a mouse model. Okay, mouse model means that you have to pay at least $3,000, right? Okay, so still it is much cheaper to work with. By the way, we in India, we have a Drosophila Resource Center, which is based in ISER, Pune. So there you can just write what fly line you want it. If any one of us in India, if we have it, we would be happy to share with you. Okay, right? Yeah. That is the good thing about working with Drosophila. So all these things fit very well with the Drosophila. And as of now, if you look at the amount of literature or the say amount of scientific concepts which have been proven in Drosophila, if you compare that with any other model organism, actually, if you combine all other model organism literature on the one side, Drosophila will be still heavier than the all other model organism combined together. Okay, yeah. So Drosophila, extensively we work on it, right? So you, as I told you, you have to choose the right model organism based on your question, and Drosophila is one of the most versatile. And of course, in the next 3 days, people will bombard you why we work with Drosophila, but I'm not going to talk about it. So the basic developmental thing, I will give you few example. This again, they will teach you a lot. So let's not worry about it. But Drosophila today can be used to study all the field in biology. There is not even a single field in biology or topic which you cannot work with Drosophila, right? So I told you the best part of the Drosophila is that the way easily we can generate mutations, right? Not only mutation, this concept, just remember it. How you will know that how normal things work, right? One way is that you should be able to label things in the normal way so that you can study the normal things, right? Say if you want fate of the cell, if you want to study, you should be label a specific cell and just follow it, that how it become eye or ear or something, right? Okay, or otherwise you should be able to make abnormal, right? We call it as a mutation, right? Abnormal and a normal. Okay, when you have an abnormality, when you understand the abnormal, then only what you are going to say is that how the normal things work. Okay, yeah. If you are going to look at this, for example, eye development, right? You need to just compare the mainland India, we call it, right? Like you guys and me. I have got smaller eyes, right? Yeah, South Asian countries, yeah, we have smaller eyes. So I'm abnormal here, right? Okay, and if you come to my place, you will be abnormal, right? Yeah, okay. Normal and abnormal is very subjective. Okay, scientifically speaking, in a Gaussian distributions, the extremes 5% are the abnormals. Okay, right? You should be able to define it. People say that how you know that what is normality. Normal means that, like say, if 100 of us are here, the 5%, if we have smaller eye, means that we 5% are abnormal, right? Okay, that's it, how you define it, right? Okay, in the Gaussian distribution, right? Okay, so if you come to my place in Manipur, you will be minority. You will belong to that 5%. So you become mutant and we become normal, right? Okay. And here at the moment, I am mutant and you are normal, right? So if we compare the genome of these two, then we should be able to figure out how many genes are involved in the eye development, which make it smaller or big, right? Okay. But of course, we need to compare it in the maximum number. And ultimately, it will be validated by looking at the genome or of the normal people, those who have got a smaller eye. Okay, there are ways to do it. Professor Ramchandra can teach you that, right? Yeah, okay, human genetics people. But the best part of the Drosophila is that you don't have to even worry about the genome analysis and thing. You can just generate random mutations which will have no eye or less eye, right? Map those genes and you can sequence in and identify the gene. Is that clear? Right? One way is that you can go from the genome in the reverse genetics, we call it, or to say you can do it by what we call as the forward genetics. And Drosophila has been a classical model to use it for the forward genetics. Today, of course, you can use reverse genetics, but the good thing about the forward genetics is that you can generate the point mutation, which you cannot do it from the forward genetics. Forward genetics, again, you have to generate this point mutation and replace the original mutation. And you don't know which one will be giving you the phenotype. But the forward genetic screen will give you the phenotype. You are going from the phenotype to genotype, so it becomes easy. Okay, and this is exactly what Drosophila people use. So if you look at all these things, I told you the leg can be developed from the antenna regions. It is the same thing, okay, right? So this happens because the antennapedia, one of the homeotic gene is suppressed, right? Ultrabithorax, which is responsible for the leg development, is expressed till the antennapedia domain. So that is how you can get the leg. And the same way, if you compare some insects have 4 wings, the Drosophila has got 2 wings. It is very easy to understand it if you work out with the Drosophila. Like here, that you have the 4 wing, you can generate the 4 wing, okay? And for the generation of this 4 wing, Edward Lewis got the Nobel Prize, okay? In other words, understanding the mechanism. Okay, and if you really want to be a good geneticist and if you want to test your genetics knowledge, please go back and read Edward Lewis this 4-wing paper, which is published in Nature. And that is the only Nature paper we know it today, which was not reviewed. In other words, which was not a referee. Because it is so complex, right? The way he has combined multiple mutation is so complex. The mechanism he has hand-drawn is so complex that if you are reading it for the first time, you will never understand it. If you understand the paper in the first time reading, it means that you are genius. It took me at least 7-8 times reading, okay? At least personally, I'm accepting it, right? Okay. And that paper, why I know it, because Edward Lewis sent it to a person geneticist in Cambridge called Peter Lawrence, just to have a look at this paper. This, I got an exciting thing. I think that I understand why few insects have 4 wings and why few have 2 wings, right? Okay. And so Peter Lawrence had a look at that paper, which is 2.5 pages printed, okay? Right, it is not a big paper. Peter Lawrence sent it to Nature editor at that time, saying that please don't send this paper for reviewing, because no referee will understand this genetics. So please publish it without reviewing. So that was the only paper in the Nature, published in Nature without reviewing it. But remember that time, people used to believe in science. People used to believe in, that somebody is conducting breakthrough science, we call it. Right? Today, no more. If you send paper and think, particularly it is from India, irrespective whether IISc or Mysore University, is still all the Western university or the referees, they will ask you more experiments to do it. Okay, right? Okay, that concept is not there right today. So you think about it, is that the Drosophila has all the mutations for all the predicted genes, all the RNAi, all the overexpression lines. You can today order it. You don't have to make it yourself. Okay, so it is available for ordering. Now, what excited me was this prize, because when I completed my master in 1994 in this university, sir knows it. I mean, I'm a gold medalist, okay, by the way. I'm proudly saying you that I was a gold medalist, but I know that I was given gold medalist because I was good in practical, not because I was good in theory. Because nobody got more marks than me in practical than theory, right? Theory, many people got more marks than me, but I got, I think, highest mark in practical. And because of that, I got a gold medal, right? I enjoy doing practical and killing flies, right? Yeah, okay. So what happened was that 1994 to 1995, that time I didn't know what to do with my life, because in Manipur, what happened is that if you are good in study, you should compete for civil service. You should become IAS officer, in other words, right? Okay. And because I was a student politician at that time, means I used to work. I mean, sir stopped me. Otherwise, I was, secretary of Manipuri Student Union, president, blah, blah, right? I was involved in everything. So my father is a politician, okay, in Manipur. So he said that you should go. You are the only son who can become IAS officer, right? I said that my heart is not in becoming IAS officer. My heart is in becoming, like scientist, right? So I took 1 year and I taught for 1 year in St. Philomena's College, where from I got my degree. Again, believe me, while teaching there in the St. Philomena's genetics, I'm a gold medalist, one of the best students in genetics, sir. Is that correct? You correct me if I'm wrong. I think I was one of the best students in genetics. And I thought that I knew everything in genetics, okay? I went in St. Philomena's College to teach genetics. And while teaching genetics, then I realized that I don't know even what is the multiple allele concept means, right? Okay. While teaching multiple allele, it's easy. The alternative form of, say, one allele is multiple allele, and you have, and you always give the, A, B blood group sample, A, B, and this, blah, blah. But actually, to teach it, it was a challenging. This is where I feel that when you are a student, please talk to your colleagues. Whatever concept you know, if you can convince your colleagues, right, then you will know it. If you are not able to convince your colleague, means that your concept is not clear. Okay, so please practice this. That is where I knew that I haven't understood genetics in that 1 year. And that made me like, at the same time, 1995, these 3 people got the Nobel Prize. Okay. And I didn't have to think twice that what I want to do, right? Drosophila people, I showed this picture, 3 people got a Nobel Prize. What more I want to do? I want to work with Drosophila, right? So it is important that an award on time can motivate you. And the award was not given to me. These 3 people got the Nobel Prize, but that motivated me to work with Drosophila. That is the truth. Of course, that time, Sir also had a grant from DST at the right time. You see, that is why I believe in Paulo Coelho's Alchemist, that phrase that whenever you want something desperately, whole universe conspires that you get it, right? And I was desperate for money because I come from a very humble background. And when I really wanted to become researchers, Ramchandra sir had his first DST grant. And I was his first student, right? So you just dream high and believe in Paulo Coelho, right? All right, yeah? Okay, so that is how I started my career. And the best few paradigm shift experiment from the simple developmental biology, I will let you know. This is something which you study every day in Drosophila, okay? Right? Drosophila has been used. Now, you think about it that in the developing oocyte, this nucleus has to be positioned on the dorsal side, remember? So that it can produce a ligand, right? Okay. Do you remember what was the ligand produced if it is in the dorsal side? Any Drosophila guy? This I'm talking about in the developing side, right? Okay. Right? Your nucleus, that oocyte nucleus is on the dorsal side. That is how it is able to repress You see, otherwise Drosophila oocyte is a completely ventralized, default ventralized. Only for the presence of the nucleus on the dorsal side, it represses the dorsal expression. Dorsal is expressed in the ventral side. Okay, so the ventral side repression of the dorsal expression from the in the dorsal side, that is how you get the your dorsal side. So ventral side is repressed in the dorsal side. Okay, in other words, just like in the human, we all are default female, you know that, right? Only when hormones kicks in because of your chromosome and the sex determining factor, males are born. Otherwise, all of us are female. Okay, in the same way, in the Drosophila, if you are looking at the dorsal ventral side is formed everywhere. Only when you repress the ventral side, right? Yeah, then you get the dorsal side in the, otherwise it will not be. So somebody tell me now, the students, don't look at the Google, because this should be in your hand like that, right? The ligand produced by the nucleus on the dorsal side. Anybody, just shout it, please. All right. This, if you love Drosophila, you should remember all this. How the ligand produced by the nucleus, right, mRNA, binds to a follicle cell receptor, right? Okay, yeah, and that represses the production of the pipes. I mean, this comes just like that. I mean, okay, so, but the question is, how people knew that the nucleus move it, right? See, I can be from Bangalore to Mysore. If you take the snapshot, you take my snap in Bangalore, you take here in Mysore, right? Okay, this is what people knew in Drosophila. But how I came, right? It is important, isn't it? You see that these are the very fundamental basic questions. But you see, I could have gone to Anandpur side in the Andhra Pradesh, right? Instead of that, I came towards here, south side, right? So it means that some directionality is important, right? And the vehicle, which vehicle I could have come by, say, car or bus or train, and all these things matters. So what I'm trying to say is that when you want to understand basic concept, you need to relate that with your everyday thing, what you do. Okay. Similarly, when people started asking questions that how the nucleus moved from the, say, posterior side to anterior side in the dorsal region, that is where people found all the mechanism of the, say, cell signaling, cell repressing, how the nucleus are pushed and everything. And you wouldn't believe it, we knew about the oocyte and dorsal ventral formation in 1980s. But this paper came out only 4 years back. What is the paper? Seeing is believing, that how nucleus is pushed. You can see that, yeah. Nucleus cannot be moving like that. So nucleus has to be pushed, because when I go from here to here, I can be either pulled or I can be pushed. Okay. All this concept, you will know it, you will appreciate it only when you see it live. That is why today live imaging technology is very important in science, because if you know how exactly things are moving, then you will understand the dynamics, the kinetics involved in the whole process. That is where biology, we are stupid, because we can't make these instruments involved in this live imaging. We, just like, you know, computer people, we can't build a computer, but we know how to use it. In the same way, the physics people, the instrumentation people, they make all these beautiful microscope where, you know, the leveling things should not bleach out, like we call it today light sheet microscopy, where you can image the live imaging for hours without losing the material, without harming the organisms, right? And to be very frank, we are one of the first to in the world. If you look at, we are one of the first to use the light sheet microscopy made by my colleague Partha Mandal in the applied physics and the instrumentation department, right? We use it to image how, say, organs in Drosophila or zebrafish develop. Okay, and this is what, so light sheet microscopy will help you develop all these things. And today we know that actually the nucleus is pushed from behind by the developing microtubules, right? Okay, I'm just throwing you. And but still, what is not clear is that okay, from the posterior part it is pushed, but how it reached to the dorsal side, not in the ventral side. So don't think that everything is known about the Drosophila. So there are unanswered questions in this, what we think that we know everything. Okay, details. So similarly, if you think about it, is that we know that the anterior posterior defining mRNA localization. By the mRNA localization, at least this you must be knowing. No? Come on. Bicoid and the nanos, right? At least, yeah, mRNA. But have you ever asked question that how these mRNAs, which are present in the posterior and in the bicoid region, in the oocyte, if mRNA are there, why they are not translated? Have you ever thought about it? You see, you are taught that these are the posterior marker, these are anterior marker. You need the nano to be moving to the dynein and the kinesin, moving it to the posterior region or the anterior region. But have you ever thought that if you have this mRNA, why they are not translated? Because it is in the cell, right? Okay. So because they are not translated, because the translation is repressed, and this is the thing what Drosophila people prove, that the mRNA, if you need to keep them untranslated, they should be repressed in a complex called as a processing bodies, right? P body. So the P body was identified using Drosophila, right? P body. In the simple concept, knowing that why these mRNA are not translated, that's all. So the one bicoid and the nanos which are not translated, you can see that they are inside. The one which will be translated after the fertilization, you will see them coming out of this complex, and you will see them as a red and the green separately. Okay. Now the question comes is that what makes it? Only fertilization makes it to translate it. If it is so, what is that? That is where the biomechanic comes, right? The biomechanic, by the way, concept from during the development mechanics is involved, cell mechanics is involved, came from the simple, what we call as the wound healing kind of thing in Drosophila, right? Okay, for that I will show you. It can be used as a model for the wound healing. But the best example came from a mutant called as the twist. Twist is a protein or the gene which is required for the mesodermal formation, all right, blastula formation. So there, people used to have a mutation in the twist, mesoderm will not be formed, embryo will not develop, embryo will be lethal. Okay, embryo will be lethal. People used to think like that. But the same mutant, if you provide a mechanical force with a needle or by pressing it, then it develops normally. Do you understand? Simple thing. The twist mutant, which is not supposed to develop, supposed to die, if you provide a needle force from outside, push it, right? Then it completes the whole development, suggesting that the whole mutation twist mutation is not able to do is that it is not able to move the cells. So if somehow you allow it to move it, then it will complete the whole development, right? That was the first evidence of how cell migration is required for the development. Okay, right? So today you think about it, Drosophila, as I told you, is everywhere in the every biology, whatever you name it, right? It is there. And we means, I don't want to say it is that, we should be beating our own drum, we would say it, but I keep on saying that the most important contribution from India when it comes to Drosophila is from this department, which was done by, of course, Professor Ranganath and Professor Ramchandra sir. I mean, I'm really proud of them. For the simple reason is that the Drosophila and NSA, which has the 2n is equal to 12, right? If you look at the chromosome evolution, right? 2n is equal to 12. If you look at the most recent evolved Drosophila is Drosophila albumicans, right? The R2n is equal to 6. Now, simple thing question, you can do genome alignment and blah, blah, blah. You can do all kind of new technology, you prove it. Yeah, you will come to the same conclusion what they did it long back using a simple microscope. That is why they are genius. Because you don't need any sophisticated tools to do science. And that is what they proved. Okay. What they did was just by looking at the chromosome squares, identifying unique property of each chromosome and trying to understand how chromosome fusion and the chromosome fission they are involved in the evolution. So the simple thing like say 2n is equal to 12 is they also be acrocentric, and 2n is into 6. All these acrocentric chromosomes should come together to give it metacentric. It's a simple concept, but how the chromosome decides that out of 12, which one I have to combine to give the metacentric, right? Yeah. So all these concepts still we need to work in cytoraces. So the cytoraces which they develop, you can see it in the evolution wise. Today, many of the cytoraces have been sequenced and things. So your my colleagues will teach you in the coming days. But what I'm trying to say is that the science is about thinking, simple things, asking questions and coming out with a simple way to do things. Instead of being cynical and say that, this happened in the Cold Spring, this happened in the Harvard, this happened in the, Oxford, we don't have it, so we can't do things. I'm sorry to say it, we don't need it. Sometimes you have to get used to your condition, pull up your socks and do whatever you are supposed to do, instead of saying that, complaining and become cynical and just say that, I'm done with it, I'm retiring, I'm going. By the way, I also will be taking very soon, hopefully, voluntary retirement and leaving science. But you see, sometimes what happens is that you have to just do it, right? Okay, so Drosophila, particularly in India today, we have more than 190 groups, PI groups. Once upon a time, we had only few people in, say, Mysore, Banaras, maybe in Kolkata, that's it. But today, all over India, we have 190 groups working on Drosophila. So Drosophila is a very versatile, asking all kind of questions. And so far, I told you that there have been many Nobel Prize. Recent one was in the 2017. And hopefully, we should have, at least among you, should be added in this if you continue to work with Drosophila and make a breakthrough. How you can make the breakthrough, I'll give you few ideas also today. And if you we talk about the EMT. That is nothing but the epidermal cell transition to mesenchymal cell. Okay, so the mesenchymal cell can move out of the cancer body cells so that they can metastasize and move to another part of the organ, right? Okay, actually, Drosophila people did it by just observing the normal oocyte development. So the what Drosophila people did was they look at this border cell. You see, this is the follicle cells. Now, this is the only oocyte which will develop to egg, right? Now, what these follicle cells from this anterior part, they move from here and come to the here in the developing oocyte. Okay, so can you play this movie? So you look at here. So these set from here and they move it here. Okay, for the movement, you need that the cell shape has to change from the columnar, which is epidermal cell like it, you know, and it changes in the shape and it moves. And once it reaches here in the anterior part, actually they become columnar again. So that happens even in the cancer. Once it reaches the target homing organs, then those cells need to de-differentiate what we call as the epidermal like so that they can grow again. Same thing happened here. Now the multiple question can ask is that why only these cells come from here? Why not from the other side or from here they are growing? What pulls them there? What is the leading cell? What makes them leading cells? What is the lagging cells, right? What makes them? All these are fundamental questions. And people have used this system to screen for the drugs which can inhibit the metastasis in cancer. There are groups, including in India, in ISAT Kolkata, they use this to look for the, say, drugs which can inhibit this movement, right? Okay, next. So it is very exciting. Today we know that at least it requires, so next. So you can see it, and today we know that there is a actin-based cytoskeleton, myosin 9, myosin 2, all these are involved in these migrations. Okay, right? If you want to just play it, it is beautiful. It is not playing, then we will move ahead. Okay, let's move ahead, right? Okay, right? Similarly, Drosophila, if you look at all the essential genes, it means that they will die. See, cancer is nothing. I'm bringing cancer because they like to talk about cancer, right? If we ask today that the students, if they come for the PhD interview, we ask them what you want to do. They will say only, we want to work on cancer and we want to work on molecular biology. Nothing more than that, right? What they don't realize is that the cancer or say muscle or the neuron, all are the name. I mean, the concept is that we use the same PCR, we use the same confocal, we use the same technique and technology, we are different. That's all. So you can choose any disease. The fundamental thing is the signaling defect, right? Okay. So it is all same, right? By the way, you are not going to cure cancer anytime soon, right? Maybe you will have antibody also, but that will be effective only for the maybe say few cancer type, but not all, because the moment genomic instability starts in the cancer, right, it is very difficult to stop it. People are dying now, CRISPR-Cas for all reversing the mutation and things, but until we have a pollution in the environment, until we have the cell division going on in our body, right, cancer is not going anywhere, okay? Yeah, right? So all those, cancer is nothing but again developmental disease, because today we know that once individual become cancerous, the developmental genes again start the embryonic For example, the one which are involved in the embryonic development in the normal condition, now the cancerous cells, they start expressing again, like say twists and all these things, right? Okay. So how you will know that they are involved? So all these things were done again by using Drosophila, by using the Drosophila wing imaginal discs, where you can make what something we call as the clones, right? The fundamental thing about the making the clone is that you can mutate only few specific cells, right? Not all the cells or organs. So the how they form the tumor will make you understand how the cancer happens, all right? And people, if you look at the north pathway, which is very important in the cancer, or let's say Hippo-Sawalder pathway, which target is the Yorkie, all this pathway, if you look at, it came from the Drosophila research, right? Okay, so I'll just run through it. So the Drosophila can be used for the wound healing, where dorsal closure can be used as how the cytoskeleton protein, they match it, all right? Similarly, as I told you, the cancer metastasis drug screening, you can make it in Drosophila, and how these cells migrated, people have now identified molecules which can inhibit these cells, stop the migration from the imaginal disc itself. So Drosophila has been used as a very simple model to study it, right? Okay, right? There are many drugs identified after the forward or reverse genetic screen, right? So these are the human diseases which are being investigated in Drosophila, and this is a website where you can identify the human disease causing genes. They are autolog in Drosophila, right? Okay, so last 10-15 minutes, yeah, okay. Jesus is telling that already I have spent more. 10 minutes, 10-15 minutes more. Okay. Exciting thing is coming, right? Yeah. That was what other people did it, right? Okay. Now we will talk about what we are doing, right? Yeah. Simple thing, right? So the thing is that you think about it, this Zolgensma is a spinal muscular atrophy treatment. This has to be given before the 18 months, right? Okay. And this is the most expensive drug available in the market. And this is possible, because of the company. Novartis is selling it, but it is not because of this Novartis company. It is because of the basic scientists. Because basic scientists work out, at what time SMA gene is expressed. If you don't have it, what happens if you have the point mutation? Because the SMA mutations are different kind. They can give rise to spinal muscular atrophy. There are 4 kinds of it. The one which we are talking here is a null. Null means the protein is not produced at it. But there are, say, for example, SMA 1, 2, this is 1, then there is a 2, 3, and 4. 4 category is the mildest, right? Okay, they survive till the adult. I mean, they completely have a normal life. But they can be given something splicing drug which can be injected, but whole thing is possible because that we knew exactly how this protein works, right? And design adenovirus which will be tissue specific. Okay, you see, I told you gene expression is very important, because you see, if you don't target the tissue, then what happens is that if you overexpress it in the unwanted tissue, right, you will be just injecting it, but if it goes to unwanted tissue, it will become off target. It will cause misexpression. It can cause some other phenotype, right? You don't want it. That is why we need to go today. We need to understand this genotype phenotype thing. But I told you that there are more than 204,000 mutations or the genomic sequences which are linked to the diseases. And these are important to know what to do if you have this mutation, okay? What we call as a informed decision, so that you can take the informed decision, okay? These are the things. But then you come here. So for the doctors, for example, say all these mutation in a protein called as the myosin binding protein C, it is the same thing. But for a molecular biologist, the mutation here will be different from the mutation in here because they give completely different phenotype. Mutation here can give you dilated cardiomyopathy, whereas the mutation here in the C-terminal can give you the what we call as the hypertrophy myopathy. It will vary in the thickening of the whether the heart muscles or the thinning of the heart muscles, right? Okay, all these things. If you know the genotype-phenotype correlation, then only you can, you know, give the drugs, possible drugs, right? Okay, right. So you need to really go back to the basic science, that how a specific transcript is present in the tissue, because today we know that the transcription can vary. Then what kind of proteome are present, what kind of protein post-translational modification are modified, so that you have what kind of protein variants are present. Then all these things are important, because all this will lead to something called as the metabolome, because metabolome is important, and this metabolome or proteome and transcriptome today can be easily modulated by what we call as the what you eat, what you do, what you think, right? That is nothing but the microbiome. Your gut microbiome can control all these things, right? Okay, so we need to go back to the basic science and understand that whatever target, what we call as the precision medicine today, we want to do it for the individual specific. We need to know in each individual what is the expression level of your therapeutic target gene, right? Their expression, their fold changes and everything. Because in every single gene, if you look at, right, their expression will change. So in what stage, right, expression is responsible for that particular phenotype, so that before the origin of that phenotype, you can intervene. Okay, so it is very important, right? And that can be studied if you have an autolog like muscle. Muscle is highly conserved. So if you are working with the muscles, then almost it follow the myoblast to the say muscle completely differentiation. We can follow it. That is why we work, right? Okay, so you can follow the genes. Then how much fold changes is present for each gene. Then what are the interactome? And because today we know that what we used to presume, at least 20 years back, we used to presume that if you know the signaling is working in this cell, this should also be working in the different cell, right? Okay, but it is not true. Today we know that the proteome interaction, transcriptome level, or how signaling work differ from a single cell to another cell, right? So you need to target cell thing, right, for the precision medicine if you are talking, right? Okay, so all these things, interactome, you need to do it, but ultimately you need to model it in the organism, and there is nothing better than Drosophila. Because for the humanization, right, let's not talk about there is not. For the humanization, whatever the mutation you have it, right, identified from your omics or everything, right, you need to put it in any model organism. And the Drosophila has got many autolog organs and tissues, and whatever you needed for the making it a transgenic, either put it the whole human gene, right, back in Drosophila to replace the Drosophila genes, right, or say making a point mutation and putting it in Drosophila. Today you can do it within 3 months. Okay, which is not possible in any other organism, right? Okay, yeah. This is where I'm talking about that you can still make future. Whatever, you want to do it today, you can run actually a company based in Drosophila or model organism, right? And we have a company, it is called as Genophy, www.genophy.com, right? That is where we make precision model for every organism, right? Okay, so I'll give you one simple example, right? I'm done with it. So we at the moment, we are working with the epilepsy, because in India, at least 9, particularly in Bangalore, up to 16 it can go. 9 to 16 individuals suffer from, every 1,000 individuals suffer from epilepsy, right? Okay, yeah. And the epilepsy is something which can be managed just like the diabetes, right? Okay, so at the moment, however, all the there are 18 drugs approved by the FDA. Out of that, 4 are given to the Indian patients, and all the 4 drugs can, if long-term intake of these 4 drugs can give rise to mobility defect called as the dyskinesia. Okay, mobility defects. So we have been looking for the alternative model because every individual is different. So we have met many mutation in Drosophila. We also can use it chemical model of induction of the epilepsy, right? So we are looking for nature inspire, or what we call as the local health tradition inspire traditional formulation, out of which what are the say compound which can be developed into drugs, right? This is what we are looking at the moment. Can you just play the movie? So this is just example, the different kind of mutation, human mutation we have made it. This is in the sodium channel SN, sodium channel 8 mutations. That is why I wanted my laptop. Okay, so even if you don't see it, this movie should have been, this fly would have been moving round and round just like many epilepsy patients does, right? Okay, and this would have been like jumping around, right? Okay, these two are different on depending on the genes. Okay, so take it from me. If you want to see it, I will show you afterwards. Now what happened is that we screen for the many local health tradition derived things, and you can see it. Just I'm showing you this example, but if you wait for 2-3 months, the paper hopefully should come out. So we have done the neurophysiology. We have gone from the Drosophila as a screening, first initial screening thing. Then we have validated today with the mice work, because you have to do it in the vertebrate, okay, higher thing. So we have done with the mice also, and it works. So the one of the compound you can see that which was not rescuing, it get rescued after feeding with that compound, right? So it get activated here and it comes down after feeding the compound, right? And this compound also work as a neuroprotective. If you feed it before, say, epilepsy happens, then it will not allow the epilepsy to develop for the 6 to 8 hours, right? Okay. And if you feed it after the epilepsy, then what happens is that the epilepsy leads to, I don't know how many of you have seen some ECG, right? Yeah, ECG, no, EEG, right? For the neuron, it is EEG, right? So the EEG, if you look at in the epilepsy, what happens is that all the neural firing will be haphazard, right? And it remains for a long time after the epilepsy bout also. So what this compound does is that it completely stabilizes the EEG after the feeding. So if the epilepsy happens, this compound alleviates the phenotype. Alleviate means stabilize the phenotype, right? So so it means that you have to take in the morning, you have to take in the evening, 8 hours, right? Okay, it works. So what I have shown you is that Drosophila can be used to study any developmental context. The basic thing is still there are many unanswered questions in the developmental biology or any ecology, toxicology, you name it, you can use the Drosophila because the whole genetics is available in your disposal, right? You have to just start. You need to go to Flybase, look at the interactome, everything, whatever you need it. If you are doing a PhD, half of your first chapter is there in the Flybase. You need to just compare, download it, everything, right? Then for your next second chapter, you order the fly from these stock centers and you start making the crosses and you analyze it, done with your PhD in 3 years, right? Okay. That is what I did in 3 years time, PhD. I think that is still a record in Mysore University, okay? But the only difference is that I slept in the lab, right? Okay, yeah. I'm joking, not saying that you people are not doing it right, okay? Yeah. So Drosophila is still a good model, and it all happened because of the I would say my mentors, particularly Professor Rangnath and these two fellows, right? They have been there in my thick time and thin time, right? Every time, if I need any support, any kind of things, moral support, I need to just reach out to them and they will give me, right? And my hardworking students and my colleagues, particularly my colleagues, because they are very critical of what I do. Without the uh, colleagues' critical comments, you can never improve your science. So that is what I believe in. And of course, thanks to Mysore University, York University, and IAC, where I've been working, right? So I'm stopping here. Thank you all. I have taken a little bit longer time, but I got excited with Drosophila and I remain excited with Drosophila, even though now I work from bacteria to human, expanded everything depending on what I want to do. But I think science should not be based on only single model organism. You should be flexible. Even though you will be working on Drosophila for the next 3 days, but depending on your science, means if you are curious about anything, just don't limit yourself, just start working on anything, right? Okay, just don't become like typical our Indian is that my wife was telling while driving back from Bangalore to Mysore that why we can't grow is that we have this thing education system where we are taught to find a job, whether it is private or the government job, we are done with it, right? Okay, but today is a time when we need to give job to our people, right? So we have to create job. We should not be depending on the government jobs or the private jobs or those who usually will give some job but also extract lots of work, right? Okay, so please don't depend on others. That is my thing. So whatever you learn in next 3-4 days, take it home and try to be independent, right? Okay, we can grow as a country, as a society, right? Okay, don't become cancerous cells. That's all, right? Yeah, thank you very much. I'm open to any question if you have any clarification. Thank you very much. Yeah. No, we should have now, otherwise the flow will go. She's telling that we should have tea. By then you would have forgotten whatever I said it. Yeah, I mean,
Hello. Good morning, sir. Sir, you have said about epilepsy as a Drosophila model.
yeah.
So which transgenic lines you have used?
So we have at the moment we have met at least 5 human sodium channel mutation, because maximum number of human mutation happen in these sodium channel proteins. So we have made it, but for anybody, you can start with a chemical treatment. In your lab, you don't have to do anything. You treat the Drosophila with the PTX, that is a compound, right? And within say 6 hours, they will start behaving like an epilepsy patient. The neurons actually fire just like in the human epilepsy patients, though. So there are something called a chemical induced epilepsy model, very well established. But in our lab, we use both chemical induced as well as the human autolog. We put back transgenically in Drosophila and we screen. Okay. At the moment, we have done it for the sodium channel 8 and the sodium channel 4.
Thank you, sir.
Any more? What happened? You people want to go for tea or you are not? You are clear, right? Yeah, please.
Excuse me, sir. You said like for getting Drosophila model flies, we can contact ICER Pune or something, right?
Yes, there is a portal, fly. Actually, you go to ISA Pune website. There is a fly Dhaka or something. What is the in Hindi name of the post? You know, post office duck, fly duck .com something. We are good in naming something, but my memory is not working. But so why, otherwise you take all the list of the people from Ramchandra sir or me. You can just email it. We have email for all the fly people. So there is a group. So whatever fly you needed, you just email to all the people that if you have it, please share with us. And we are good. We are not greedy or selfish. We just share if we have it. Okay. So those conditions, you don't have to pay anything. You have to just pay. I mean, we will send you for free. But if you want other flies like say transgenic, something which we don't have, from Bloomington, we order once or twice in a year from IAC. So you can give us at least around say 10 or 15 minutes, we can accommodate it. So you see each line is $26, $27. So we pay that. So you at least we can get you at least 3, 4 or up to 15, depending on how much money is left. We can order it for you and we can supply.
Thank you, sir.
Any other? Right. If there is no question, thank you very much. Right. Thank you. Yeah.
All the participants here to our fly group, so, Kaushik will send all the email.
No, this fly group is only for the PIs. This the one which Lakotia has sent out is only for the PIs. Yeah. So if any PI, if you want to use fly as a research thing, then we can include your name. Yeah. Right. Thank you very much. Yeah.
Sir, to inaugurate the scientific sessions, we have a demonstration. So I request NBR sir also to please come on the dais. Flight transfer. To inaugurate the scientific hands-on training, right, sir? So we are doing hands-on inauguration. Yes, sir. Yeah. So this is how we transfer the flies from one bottle to the other. So we'll have to tap.
So you can see there is no fly left on. This is how we do in India, but if you go to Germany, you are not allowed to transfer like this outside. So you have to work within the lab environment because nowadays they think that Drosophila, even though it is, we call it, it will not harm anything. They are afraid that the transgenic fly, however, will contaminate the water circulation. They don't allow to transfer the fly outside.
Thank you, sir. Thank you so much. Thank you. Thank. So we will break for tea. So after the tea, we will be shifting to Department of Genetics and Genomics. So after that, the post to which all the sessions will commence in the department. Thank you so much.